Good morning, students. Today we are discussed about ammonium assimilation in plant physiology for MSc final students or BSc second year students also. Ammonium assimilation. Plant cells avoid ammonium toxicity by rapidly converting the ammonium generated from nitrate assimilation or photorespiration into amino acids. The primary pathway for this conversion involves the sequential actions of glutamine synthase and glutamate synthase. We will discuss the enzymatic process that mediate the assimilation of ammonium into essential amino acids and the role of amides in the regulation of nitrogen and carbon metabolism. Converting ammonium to amino acids require two enzymes. Glutamine synthetase GS combines ammonium with glutamate to form glutamine. Ferredoxin reduced in and convert into ferredoxin, and it forms in the presence of nitrate reductase. It forms heme and separated converts nitrite into nitrous oxides or ammonia. Glutamate plus ammonium plus ATP, it forms glutamine, ADP and inorganic phosphate. This reaction requires the hydrolysis of one ATP and involves a divalent cation such as magnesium, manganese or cobalt ion as a cofactor. Plant contain two classes of GS glutamate synthetase enzyme, one in the cytosol and the other in root plastids or shoot chloroplast. The cytosolic forms are expressed in germinating seeds or in the vascular bundles of roots and shoots and produce glutamine for intracellular nitrogen transport. The GS in root plastids generate amide nitrogen for local consumption. The GS in shoot chloroplast re-assimilates photorespiratory ammonium. Light and carbohydrate levels alter the expression of the plastids form of the enzymes, but they have little effect on the cytosolic forms. Elevated plastid levels of glutamine stimulate the activity of glutamate synthase, also known as glutamine 2 oxoglutarate amino transferase, or we can say GOGUT. This enzyme transfers the amide group of glutamine to oxoglutarate, yielding two molecules of glutamate. Plant contains two types of GOGUT enzymes. One accept electron from NADH, the other accept electrons from ferredoxins. Here is the equation where glutamine and GOGUT plus NADH, it forms glutamate and NAD. And in other, glutamine plus Gogat enzyme plus ferredoxin in reduced form, it forms glutamate and ferredoxin is oxidized. The NADH of enzyme Gogat is located in plastids of non-photosynthetic tissues such as roots or the vascular bundles of developing leaves. In roots, NADH Gogat is involved in the assimilation of ammonium absorbed from the rhizosphere. The soil near the surface of the root is also called rhizosphere. In vascular bundles of developing leaves, NADH and Gogat assimilates glutamine translocated from roots or senescing leaves. The ferredoxin dependent type of glutamate synthetase is found in chloroplast and serves in photorespiratory nitrogen metabolism. Both the amount of protein and its activity increase with light levels. Roots, particularly those under nitrate nutrition, have ferredoxin Gogat in plants. Ferredoxin Gogat in the roots presumably functions in incorporate the glutamine generate during nitrate assimilation. An NADH dependent form of GDH, glutamate dehydrogenase, is found in mitochondria, and an NADPH dependent form is localized in the chloroplast of photosynthetic organs. Although both forms are relatively abundant, they cannot substitute for the glutamate synthetase or and Gogat pathway for assimilation of ammonium, and their primary function is in deaminating glutamate during the reallocation of nitrogen. Transamination reactions transfer nitrogen. Once assimilated into glutamine and glutamate, nitrogen is incorporated into other amino acids via transamination reactions. The enzymes that catalyze these reactions are known as aminotransferases. An example is aspartate aminotransferase. Which is called ASP and AT. Aspartate aminotransferase, which catalyze the following reaction: glutamate plus oxaloacetate plus aspartate, and it forms aspartate and 2-oxoglutarate, in which the amino group of glutamate is transferred to the carboxyl group of aspartate. Aspartate is an amino acid that participates in the malate aspartate shuttle to transfer reducing equivalents from the mitochondrion and chloroplast into the cytosol, and in the transport of carbon from the mesophyll to the bundle sheet for C4 carbon fixation. All transamination reactions require pyridoxal phosphate, which is called vitamin B6, as a cofactor. Aminotransferases are found in the cytoplasm, chloroplast, mitochondria, glyoxysomes, and peroxisomes. The aminotransferases in chloroplast may have a significant role in amino acid biosynthesis because plant leaves or isolated chloroplast exposed to radioactively labeled carbon dioxide rapidly incorporated the label into glutamate, aspartate, alanine, serine, and glycine. Asparagine and glutamine link carbon and nitrogen metabolism. Asparagine isolated from asparagus as early as 1806 was the first amide to be identified. It serves not only as component of proteins but as a key of compound for nitrogen transport and storage because of its stability and high nitrogen to carbon ratio. The major pathway for asparagine synthesis involves the transfer of the amide nitrogen from glutamine to asparagine. Asparagine synthase, the enzyme that involves the catalysis, the reaction is found in the cytosol of leaves and roots and in nitrogen fixing nodules. In maize roots, particularly those under potentially toxic levels of ammonium may replace glutamine as the source of the amide group. High levels of light and carbohydrate conditions that stimulate plastid, GS and ferredoxin Gogat inhibit the expression of gene codings for aminosynthetase and the activity of the enzyme. The opposing regulation of these competing pathways helps balance the metabolism of carbon and nitrogen in plants. Conditions of ample energy stimulate glutamate synthetase and Gogat and inhibit aspartate synthetase. Thus, they favor nitrogen assimilation into glutamine and glutamate compounds that are rich in carbon and participate in the synthesis of new plant materials. In contrast, energy limited conditions inhibit GS and Gogat, stimulate asparagine synthetase and thus favor nitrogen assimilation into action into asparagine, a compound that is rich in nitrogen and sufficiently stable for long distance transport or long term storage in plants. Thank you, students. Tomorrow we will discuss about the new topic amino acid biosynthesis in our next class. So thank you for today.